So good afternoon, everybody. It's a pleasure to be here. This seems to work. Excellent. Yeah, so it's a pleasure to present you a bit on our work on machine learning in personalized medicine. I'm not giving an overview of the whole field. I'll just focus on exactly how we use these technologies to try to improve patient treatment and patient outcomes. And we do so, and I'll show you more details, using generating these kind of images of patient tissues in high throughput. So I'm an assistant professor at the ETH Zurich, and my lab is the Snijder lab. Good. So why do we need precision medicine? Despite amazing advances in patient treatment, it's clear that there's still a lot of, for instance, tumors and other diseases that kill patients on a devastating rate. And one example here, I'm showing the survival, so how long patients diagnosed with leukemia live. Okay, my connection is a bit slow. I'll skip the pointer. And what you see here is in the blue line, patients for which a certain mutation were detected. Is everything okay? Or? Yeah, okay, but it won't share on Zoom. But so what you see in the blue line is the survival curve of patients with acute myeloid leukemia that have a FLT3 mutation and that have received targeted therapy specifically designed to interfere in that driving mutation in this disease. And that does better than just chemotherapy for these patients. So it is a really significant improvement. Nonetheless, within 2 years, most of these patients have died, right? And that's more or less the current state for acute myeloid leukemia. So what we've set out with my lab is to say, okay, we need to find better ways. We need to find better ways to identify personalized treatments for cancer patients. And what we've developed in this context is a very simple process that says, okay, can't we just take a small tissue from these patients and test how all of the possible treatment options might work on that patient's cells? And we just do that in the lab with fresh patient tissue. So the process is actually quite straightforward. Peripheral blood or bone marrow or skin or tissue biopsies reach the lab. And then we take these cells and put them in so-called 384-well high throughput drug screening plates. So one such plate has 384 small chambers, and in each chamber we can put a different drug or drug combination, something that these patients might receive in the clinic. And then we incubate these cells, so the cells live together with those drugs, and these are mixtures of cells, right? So we'll get healthy patient cells, cancer cells from these patients. And then typically after 1 or 2 days in the incubator in the lab, we then fix and stain all these cells with fluorescently labeled antibodies, allowing us to see what kind of cell each cell is. And we image that with automated microscopes, producing really large-scale image-based data sets. And just to give you a little bit of an overview of the work in the lab, we use this for 2 sort of different applications. One is really systems oncology and personalized medicine, including running clinical trials where patients basically are treated with the drugs that we see work best for them. And in this context, the concept is always simple. There are some cells that are cancer cells, and we want to find a drug that specifically kills those cancer cells. But we also do sort of more immunological focused research with this technology. I won't talk about this topic today. Okay, well, it's getting there, I guess. Yes. So simply put, what it means is that we can, within a few days from a small blood draw, so this is just 9 milliliters of blood as it's taken on a really routine basis, produce such, and this is very zoomed out data, high throughput functional perturbation data, where for the cells from that small blood draw, we can measure those 384 different responses or perturbations, including possible treatments that this patient might receive. The original data, though, is, of course, much higher resolution than I'm showing here. And I apologize for the speed of my connection. The original resolution is really this kind of single cell, rich, multicolor immunofluorescence data, where each blue dot is a single nucleus of a cell. So a nucleated cell contains the genome. And then the different colors and morphologies indicate different kinds of cells, allowing, and so the immunofluorescence, this kind of staining process, is really important to identify, what each cell type is. So what we do in the lab is we stain these cells with different antibodies. I'll skip over the details because it gets rather experimental. But we have different colors that we can basically attach to different kinds of cells. And critically, we then come in with convolutional neural networks that we've trained across large-scale data sets that look at each individual cell and tell us what the identity of that cell is. Was it a cancer cell? Was it a T cell, a B cell, an immune cell, or other cell types? And in this example, we've trained a single convolutional neural network, a ResNet architecture across some 250,000 manually annotated cells in 8 different cell classes. And then, for instance, test its performance on a validation set that 48,000 cells manually annotated that the neural network never saw before. And in this particular example, this is peripheral blood cells, like we have it all floating in our veins. We can get a single convolutional neural network that is 98% accurate across images of such cells from 150 different healthy donors. And those healthy donors are processed at different days and different times. So, you know, that there's a lot of experimental and batch effects, variability in that background. But these neural networks can really be trained to be robust to that kind of technical variability. Now, that was a very large-scale manual annotation challenge, of course. There were people clicking a lot on those cells, giving them labels. In essence, in parallel, we've realized that for this kind of data, you don't actually need all that supervised training, even unsupervised neural networks, such as here, self-supervised dyno-trained vision transformers, can do really excellent in a fully unsupervised way, just sort of staring at thousands of such single-cell images and then clustering them based on their similarity. And you find that the nearest neighbor, top 1 accuracy, treating this as a classifier, is in fact 95% accurate on the same 8-class challenge. So now when we know what is the identity of each cell in each of these images, and then across all of those many images, we can turn this into synthetic data sets or sort of the computational vision of each, analyze the class, the abundance of each cell type, its spatial interactions, do network or graph-based analysis on these pictures that we generate, but also just have a simple efficient counter in each condition, how many of each cell type is there alive and which perhaps died in response to a treatment. So the early application that we developed here was to actually guide patient treatment with this platform. And we've been doing this now for over 10 years. So what it means is this is a PET CT scan of an elderly man who was at the time back in Vienna, where I did my postdoc, with a diffuse large B-cell lymphoma, so really devastating lymphoma type, had received all the standard of care treatments, at which point the clinicians came to us and said, hey, here's a small biopsy from this patient. Can you not tell us what might work for this patient's tumor? So we took the cells from that small lymph node biopsy, screened 104 possible treatment options, and then prioritized them a few days later based on which drugs most specifically killed that patient's cancer cells. And what we found was that out of the 104 treatment options that we screened, only 6 significantly specifically killed that patient's cancer cells. So we reported that back to the clinicians, that, hey, look, we've tested this biopsy. There were only these 6 drugs that seemed effective. And the clinicians were excited because the second best drug that we found in our screen was a drug that made a lot of sense for this disease. It's called ibrutinib. It's a BTK inhibitor. So the patient went on ibrutinib treatment and had a long-lasting complete remission to just that one drug. Another early such example back in 2016, middle-aged woman also with a lymphoma. You get this tumor buildup around the lungs. We could screen hundreds of possible treatment options. The clinicians again picked 2 of the top drugs out of our hits, and the patient had a very strong partial response to that treatment. So by now we've run this platform through 2 different clinical trials. These are both interventional studies where patients systematically get treated based on our recommendations, and we followed them for years to see how well they respond to their treatment. One study was called the EXALT-1 trial. 52 patients received what we call pharmacoscopy-guided treatment. And what we see is that a quarter of those patients had really long-lasting progression-free survival, up to multi-year benefit from this functional precision medicine, as we call it. And here, by the way, we compared that to the prior treatment response of that same set of patients. So how well did those patients respond to the previous treatment line? And we see that we do significantly better than that. In parallel, in Switzerland, we ran another such study, specifically on leukemia patients. And what we see is that patients that received pharmacoscopy recommended treatments on average lived 3 times longer than patients that receive treatments that weren't recommended by the platform. So these are both not yet randomized, but at least highly encouraging signals that it is feasible and in fact effective to guide patient treatments for cancer patients with such a functional precision medicine platform that really in the background, it's a big image analysis pipeline with quite a lot of machine learning in the background. So one of the things we've done over the past years in my lab in Zurich is say, okay, these were all blood cancers. Blood cancers in a way are simple because you don't need to dissociate the tumor. There's a lot less questions about, solid tumors and metastasis. If we take a small biopsy from one side, does it reflect the response of the whole patient? And one of the most advanced studies that we've done now on the solid tumor space is to adapt this drug screening approach that we call pharmacoscopy to one of the worst cancers there is, called glioblastoma. So these are the most aggressive primary brain cancers that arise. And this was work that was just published in Nature Medicine 2 weeks ago, led by the amazing Soyan Li and together with the University Hospital Zurich. And so one slide about glioblastoma. It really is the worst primary brain cancer upon diagnosis. Till today, patients survive less than 2 years. The treatments haven't changed much for the past 20 years. It's surgery, radiotherapy, and chemotherapy with a drug called temozolomide for most of these patients. There's real challenges. The tumor is protected by the blood-brain barrier. So if you take a pill, the drugs don't reach the brain most of the time. And the tumor produces an immunosuppressive environment. So also immunotherapies thus far have not made a big impact for the treatment of glioblastoma. And the tumors are thought to arise from glial progenitor cells, but the genetics and the biology is really rather complex. So the process we've set up here is the same, pretty much the same as for the blood cancers. After surgery, we receive tissues from the patient in the lab, dissociate these tissues and distribute them in these 284-well drug screening plates, incubate the cells with drugs for 2 days, and then fix, stain, image, and analyze this large-scale image-based data sets. So we're maintaining sort of all of that tumor complexity. All the cells that were in the biopsy or in the surgery are now in our wells. And we can identify a subset of cells that are sort of so-called glioblastoma stem cells or glial stem cells that we think are the causative subpopulation of cells in this disease. And again, we control for toxicity by also trying to keep alive the healthy patient cells. And this process is fast and automatable. And to our surprise, when we started to generate these data sets and process this glioblastoma surgery material, again, we could see that we can produce these kind of 284-well high throughput drug screening plates with such fresh patient material. But the images look strikingly different than anything we've seen before in the blood cancer space, including for some patients, these glioblastoma cells here in yellow and green, producing these really elaborate cell networks in our wells. So there's only one drug that's really relevant for these patients in the clinic. That's temozolomide. It's been around since, it's been standard of care since more than 20 years now. It's a chemotherapy. So one of the first things we did was in some of these wells, we put that drug and then we screened that across patients and we asked, okay, patients for which we see the drug work, did they do better in the clinic? And that is very much the case, both in 2 different patient cohorts. We see that patients that respond well to the standard of care chemotherapy in vitro in our assay also go on to have better responses in the clinic, including longer progression-free and overall survival. And inversely, if you don't respond to the drug in our well, then in our wells or in our assay, these patients die faster. But of course, we have a high throughput drug screening platform. So it's not just one drug we test. We have these hundreds of wells we can screen for new things in. And one of the things we did was, and this was really Soyan's work, to collect this library of blood-brain barrier permeable drugs that people take on a daily basis around the world. So these are what we call neuroactive drugs. Think of antidepressants, antipsychotics, anti-Parkinson drugs, anti-nausea drugs, drugs that are all designed to pass the blood-brain barrier and do something in the brain. So we collected 70 or 69 of those, and we ran them through the surgery material of 27 patients, all freshly collected over the past years in Zurich. And of course, we know a lot about these patients, their genetics, their sex, their history, et cetera, their clinical outcomes. And to our surprise, what we saw was when we started to screen this, and this is very different for different tumor types, but in glioblastoma with this library in these patients, what we found was that there was a limited set of these drugs that are otherwise clinically safe and approved that showed quite strong efficacy here in blue with stars on a subset of these patients. So it's really highly encouraging. It suggests that the top one here, vortioxetine, is in fact a clinically safe antidepressant. So it suggests that there might be drugs that we can repurpose for the treatment of glioblastoma. And then one of the fun things, and again, this is very much a precision medicine effort, is that even from our primary screening data, we can come up with hypotheses of why some drugs work for some patients. So we see drugs that work specifically for men, but not for women. We see drugs whose responses are particularly happening in patients carrying certain mutations. But for instance, also drugs that work better for the elderly, or here, vortioxetine working better depending on how the morphology of the cells looks like. But we were hoping that there was somehow a simple mechanistic story coming out of the screens. That turned out not to be the case. So our top hits, these drugs here on the left that seem to work for some patients, they're not statistically significantly different from all the drugs that didn't work. Some antidepressants worked, most antidepressants didn't. Some antipsychotics worked, most antipsychotics did not work. So there was no simple mechanistic insight coming out of this. And that's the last bit of machine learning I just want to show you for today. We then said, okay, but this is sort of the simple mode of action classification of these drugs. Of course, we know a lot more about what these drugs do. And in fact, there's whole large-scale databases out there that describe what proteins these drugs might in fact interact with. So we took all the drugs that we screened, and that library was a bit bigger than what I've shown here. So in total, some 130 drugs that we screened across these patients, annotated them for sometimes working or never working in these patient samples, and then collected a bunch of data on these drugs, including what proteins have they ever been reported to interact with, biochemical assays and functional assays. Here in red, I hope you can see it. And then we said, well, that is just the first layer of information, but we can expand this by also annotating what proteins those proteins themselves interact with. And that is the second layer of information that we collected for this data set. And the concept we came up with is something we call CoStar, and that is a very simple question. It's from this sort of network, are there second-degree drug targets, so second-level protein interactors for these drugs that might significantly stratify which drugs work and which drugs don't work? So unique secondary drug targets associated with hits, and perhaps secondary drug targets associated with non-hits. And we do this with very simple binomial loss of regression, just to get that minimal number of secondary drug targets that is maximally predictive for whether a drug works or doesn't work from this giant hairball. And our model training data looked pretty reasonable. So a simple, small linear model based on this network of data could predict 20 of our test data drugs correctly with only a single false positive. And the fun thing of doing this with something as simple as regularized regression is that you get a very interpretable and straightforward model. So we can look at what features this model has picked up on, what in fact are those secondary drug targets. But we realized we could also do something else, and that is just run this simple linear model over all the chemical space that has been annotated the same way. And that's what I'll show you first. So we then collected sort of this network data, not just on the 130 drugs that we experimentally screened, but we collected it over 1 million compounds that have been annotated in databases out there. And then just ran this simple linear model over that giant network and asked the model, which of these drugs would you predict work well and which do you predict don't work? And then we went back and commercially bought a number of those positively predicted drugs and negatively predicted drugs. We just screened those through more patient samples to see if what the model had learned was in fact predictive. So here, 4 rows, 4 new glioblastoma patient samples. We have our positive controls, negative control. Showing you the results for the predicted non-hits, basically showed not a single drug having any efficacy and several drugs showing in fact toxicity in these samples. And to our utter surprise, the predicted drugs were massively enriched again for on-target significant anti-glioblastoma efficacy across these 4 patient samples, with a total area under the receiver operating curve of around 0.94, suggesting this is in fact a very predictive classifier. And then the other beauty is we can just look at the biology that this model predicts, and I'll spare you pages and pages of results, but this prediction turned out to be very much true. So in pink here, these neuroactive drugs that worked in our assay, they'll trigger Jun activation, AP1 transcription factor pathway activation, leading to upregulation of anti-tumor or tumor suppressors called BTG1 and 2. A little last nugget of validation for this data. So the strongest drug that we found from this effort was vortioxetine. This is a safe and clinically approved antidepressant that's taken by millions of people around the world for months. And so what we did together with our clinical collaborators was a number of in vivo experiments where we inject glioblastoma initiating cells into mice, let the tumors grow, and then start treating these mice, either with a standard of care, negative controls, positive controls, or this vortioxetine. And what we found to our surprise is that this safe antidepressant is as good as temozolomide, which is known to work in these assays in vivo. So it prolonged mouse survival significantly and as much as the chemotherapy, even though it's at a lower concentration and a much safer agent. And when we then combine these drugs in mice, because we see that they have very complementary mode of actions, we see that the combination of the antidepressant plus chemotherapy starts to rescue mice and have additional survival benefit. So really, by all means, this vortioxetine, which we identified, seems to be an attractive drug repurposing candidate for this terrible glioblastoma, these terrible brain cancers. And I'm happy to say that the phase 2 trial to validate this finding, to see whether vortioxetine actually is beneficial for these patients, is now in preparation and hopefully starts within the next months. Yeah, so I'll keep it short. I'll keep it at that. I'd like to acknowledge the collaborators for the glioblastoma work, Michael Weller and Tobias Weiss at the University Hospital, but perhaps more importantly, the team that has done the work, including Soyon, Clara is here, by the way, work not shown, and others. So I thank you for your attention. I'm happy to take questions.
Yeah, I think we can take time for questions. There are already a few.
Okay, thank you for the talk. Excellent topic and fantastic theme. But my question is like this. So antidepressants are one of the most prescribed drugs in the world. And I assume that cancer patients receive them a lot, obviously. So how did it happen that this increased effectiveness of cancer therapies together with antidepressants wasn't caught earlier from practice? I mean, it should be obvious.
Yeah. It would be nice if it was obvious. It's not obvious. So there's a few factors. We dug into this quite extensively. So this kind of real world data, can you not see that patients that take, or people that take this drug develop less glioblastomas? And inversely, perhaps you have glioblastoma patients that take the drug and do they do better? So we've dug into American data, Korean data, and Swiss data, and the overlap, the coincidence of it happening is too low. So vortioxetine is approved as third-line antidepressant. That does mean millions of people take it. But glioblastoma is a really rare tumor. So in essence, you might catch 1 or 2 patients that take in the drug and have it, or the other way around. And then the heterogeneity of the data is crazy. So patients might have taken vortioxetine before and then later on developed glioblastoma. It's really not easy you really need large scale data to have a convincing answer. There is real world evidence on other antidepressants, but it's conflicting. And we think it's yeah, anyway, so it's inconclusive. And in the end, you just need to run the trial. Yeah.
Can you say a little bit more about why this works and how did you find that explanation? Because you're using neural networks and it's not immediately obvious how to arrive at the explanation.
So the CoStar algorithm, it's really just linear regression. So we just generate a minimal set of non-zero coefficients that you can attach to secondary drug targets out of this hairball that are then predicting whether a drug works or not. So there are a few sort of secondary drug targets that add negative evidence, and they say, well, look, if a drug has that as a secondary drug target, it's repeatedly seen to not work. And by far the strongest positive signal was in fact this BTG. This gene here. So if a drug by any means has a secondary drug target connection to this gene just got a really high coefficient, and it just helps predict that that drug was working. So here, it's really not a neural network. We take a biologically motivated network and apply simple linear regression, although neural networks would have picked this up as well. But the interpretation is that much straightforward with a linear model. Yeah.
One more question here.
Thank you for the great talk.
Thank you.
My question is regarding using blood-based biomarkers for such things, because taking, for example, tumor samples is a rather invasive procedure and it's not quite easy to implement in every case.
Yeah.
So is there any work being done on that?
Yes, absolutely. So circulating cell-free tumor DNA, circulating tumor DNA as one actively rising biomarker from less invasive biopsies is definitely a big field. And there are a lot of sort of non-cellular but molecular biomarkers that are used in clinical routine. So that is very much the case. They're most of the time used just for diagnosis. Is there a tumor? What kind of tumor? And they might not per se tell you how to treat that tumor. So with the circulating tumor DNA, you now can start, you see companies and sort of in clinical routine, genetics being done on that. So you can detect possibly some or some mutations of that tumor. And that might tell you, what targeted therapies might help. But our essay is really not about, so most of the patients that we're looking at are patients for which genetics is not helpful at all. So either they've had already received targeted therapy and then relapsed, or like in glioblastoma, there is just no targeted therapy. So there's complementarity there.
From what I understood, the accuracy, the effect of the therapy depends on the accuracy of the biomarkers that you identify. Do you have an idea if you can expect better and better effect if you identify more markers and getting a better and more exact profiling of the single patients?
Yeah, so we do take multiple strategies here. We do find that the assay, this functional assay itself, can become a biomarker to stratify patients that might benefit from the treatment and that might not benefit from the treatments. Those interventional trials that I showed are in essence that, taking a functional profile as a treatment response decision. But of course, and like in the glioblastoma world and for general expansion, it was really great if we can pinpoint who benefits from these drugs based on simple routine markers. And even there, we find that this kind of screening is a really efficient way to identify such biomarkers, because you can both scale up the dimension of drugs you test and immediately screen that across patients. So it becomes a really efficient way to detect those biomarkers, or at least biomarker hypotheses that you can then validate. And in the end, we have projects where we integrate DNA, RNA, protein level information. Depending on the drug, the disease, the stage of the disease, the type of biomarkers that we find predictive are really varying. So they really come across different data modalities. And I think it's, in that sense, the future for precision medicine would be multimodal, right? You cannot just have a single assay that detects some biomarkers, but you really need to start measuring across data modalities to come up with optimal treatment decisions.
Okay, I don't see any further questions. So thanks again.